Welcome, everybody, to this workshop from AG2PI. The workshop title is Genotype to Phenotype for Non-Biologists, or as we like to call it, G2P4 and B. We love acronyms here. So hopefully you're in the right place. The purpose is to provide introductory material on genetics and genomics for non-biologists, and especially those that are interested in applying their data science skills to the genotype to phenotype field, or the genotype to phenotype question. As biologists, we recognize that this cannot be a replacement for a course in genetics or genomics, but it hopefully will give you a view of the field that can help you understand questions of interest that we have in genetics, and that hopefully you're interested in applying your skills to. And really emphasizing the complexity that's present in the biological samples that we work on. So I will go through sort of a quick summary of what's going to happen in the workshop schedule. There's 4 sessions, 4 days, in which we'll have 2 sessions per day on Thursdays for the next 4. And I'll go through these in a little bit more detail, but essentially we'll have 2 periods per day. And the time frame is from 11 to 1 Pacific, 2 to 4 Eastern, 1 to 3 Central. A little bit of detail on the workshop structure. The idea is we're meeting on Zoom, and then Nicole will create breakout sessions a little bit later in the hour. And the idea would be to have a general 30-minute lecture, which is what I'll give here in a little bit, with then followed by breakout sessions where you guys can discuss some of the questions that I'll pose and the speaker will pose at the end of their half hour. And then do that for about 15 minutes and then come back together with 15 minutes worth of discussion to either continue to discuss those questions or to answer other questions that you might have presented. And you'll have the opportunity in the Zoom chat to ask questions about content. And if there's a question that we catch that we need to answer during the presentation, that's fine. I'll try to do that. But in addition, we'll have that very last 15 minutes to discuss those questions as well. Ryan is also organizing this series of lectures and this workshop. And so he's going to talk to us about ways to use a particular functionality at HackMD to answer and discuss questions on the breakout. So I'll escape here and let Ryan take over.
Thanks, Chris. So if you are unfamiliar with hackmd.io, this is a sort of a dynamic chat, like a persistent, like a notepad. And so I dropped the link in the Zoom chat right here. And so we'll have one of these for each of the lectures for the breakout questions afterwards. So let me just walk you through this introductory one right here. So there's a link included. If I click on this it'll just look like a website. There's a tutorial on the hackmd.io page I linked in the chat, which tells you some of the ways that you can utilize HackMD. But what we're interested in mostly is the split pane view. And so at the top of each of these, there'll be the AG2PI logo, what the lecture is for that particular section, the learning objectives, and then there will be pre-populated questions. And then when we randomly assign you to breakout rooms, we provide some space right here. So you can just scroll to your room number that you're in, designate one person from your group. And then when you're in there, you can answer these questions by just going to this line after the colon and hitting enter. And then you can push tab and it'll move over and the star will change to red. And now I can as an example, say this is breakout room one's answer to question 1. Another answer to question 1, etc. For all of the groups. Everybody should have edit access to this. So when we break out into the breakout rooms and we're answering these different questions, you can scroll through. There'll be little dividers between question 1 and question 2. You can just find those breakout room numbers, answer your question as a group, and then we'll come back together and discuss it. After we're done, with the workshop, I'll lock the write access to it. So this will be unchanged afterwards. So all of our responses will be recorded, and then we can view them afterwards as a website. It'll just look like this and be indexed on Google. So that is all that I have to say about the HackMD. Does anybody have any quick questions about it?
Hey, Ryan, could I just quickly jump in with one comment?
Yes.
Yeah, so just one of the things that we want to make sure that people appreciate is especially if you're coming into biology, you have a background in engineering or data science, or computer science, or other aspects. One of the key things that really is helpful in terms of starting to work across these domains is developing the common vocabulary. And if there's one thing that biology is great at, is having a lot of terms that are very complex, that often don't mean what you think they may mean in typical language. Transcription and translation are 2 words, for example, that have multiple meanings and very specific meanings in biology. And so the overall goal of this is to really increase the ability to communicate across these disciplines. And so even as you're in these breakout groups and you're working on these questions and you're thinking about things, if there are terms that you're confused about, if there's concepts, these terms often represent some biological phenomena. This is a great place to post those, and so that the entire AG2PI team can be aware of them, and the speakers can highlight those as discussion topics during the more informal breakout sessions when we all get together to chat about different things. So I just want to let people know that those you can answer questions, you're not going to get scored on them, but it's a great place also to highlight areas that you may be confused, either in terms of terminology or in terms of concepts.
Great, thanks for that, Eric. I'll go ahead and go back to my slides. So what I'd like to do for the time I have remaining is to first go through a very short description of what we're going to cover over these next 8 sessions, and then spend about 10 or 15 minutes just describing a couple of overviewing sort of the fundamentals and really emphasizing one particular aspect of what we mean by simple and complex genetics. So genetics is the study of how information flows in biology. To really understand genetics, we need to embed many concepts on several levels, because biological information and specifically genetics is embedded in many different levels. We can talk about populations, the genetics of population, and how genetic information, biological information flows through the populations by looking at crossbreeding, for example. We can look at the genetics of individuals. We can look at how genetics controls how tissues, cells, and molecules interact. So genetics is very complex. It really demands your attention because information is interactive, and you see that genetic information tracking through all of these levels. So about that in general, give you some overall view. And then in session 2, Cliff Wheel will talk about molecules, central dogma, and phenotypes, and really get into the details of what these molecules are doing and how they really impinge on function. So cells and organisms are very busy, they're complex, and they really are 4-dimensional. There's a lot going on with many different things happening. And the cells need very clear instructions and a lot of communication and coordination. So the central dogma DNA being the repository of information, which is then transcribed into RNA, and then the RNA is translated into the functional unit, or what we would normally think about as the primary functional unit of biology, the protein. And so the general concept of genotype and phenotype are central to what we're talking about in AG2PI. So he'll also talk quite a bit about what a genotype is, the complete set of genetic information within a cell or organism, and the phenotype, any outward manifestation of that genotype.
Hey, Chris, can I interrupt real quick?
Yep.
Can you share the presentation mode? We can see presenter mode and it's making the slide a little small.
Oh, okay.
Thank you.
Is this what you see? Do you see?
I see the presenter mode still. So I can see all your slides on the left.
Yeah.
Okay. Let me try it this way. Is that?
There you go.
Perfect. Thank you.
Okay. I know when I moved it to my laptop so I could look directly at the slides, then it changed on me. But at any rate, so then Clifford Wheel will then have another session in which he'll take the molecular information and then expand that and get you to think about alleles and inheritance and linkage. So in terms of somewhat more about how these molecules actually are where they are and how they actually transmit from parent to offspring. So alleles are the form of a gene. So just another technical term, but basically genes come in very different, in multiple different forms, which may have different functionality. And so we need to follow those different forms, and we call those alleles. We often use genetic alleles to tell us something about that gene function. And organisms that we primarily deal with, eukaryotes, have 2 copies of every chromosome. We also often call that diploid. Chromosomes are keeping track of how many chromosomes are present is important because there's concepts of gene action and dominance and recessivity, and he'll talk about that. But they segregate from one another. And so you have parent to offspring creation of new entities with each chromosome from each parent coming back together again. So there's aspects of really following where all these alleles are going. And then a little bit more about the location of alleles relative to other genes. Alleles of one gene might be located on the same chromosome. And if they're located closely enough, they often are inherited together on the same actual chromosome. But if alleles are on different chromosomes, they may or may not be inherited together. And we call that, and so they're independent from each other. We call that unlinked. In the 4th session, Ryan will join us and give a presentation on quantitative methods. And so we can then think about how do we actually do quantitative and statistical approaches. There's a lot of complexity, as I mentioned before. So one of the things that we have to do is apply mathematical tools to really describe population and describe even molecular interactions. One of the things that we like to do is quantify differences in genomes. So one way to do that is to analyze the structure of the genome all the way down to the individual units, the individual base pairs. And we then identified variants, and we call those single nucleotide polymorphisms. So variants at the single base level. And once we have that most defined information, the genetic variant information, we can use that in describing how those variants are transmitted from parent to offspring that we can measure phenotypes in. So we can observe phenotypes and try to correlate between the DNA sequence variants that are in those individuals and try to correlate the presence of those variants with observed phenotypes. But we can also then begin to think about how the individual organisms are interacting with the environment, and does that change how the genome is functioning? So we can look at the responses of cells and organisms to the environment, and we can quantify that at least one way to quantify that is with what's the activity level of the gene. And we often call that the gene expression study. And then I'll come back in session 5 and talk a little bit about epigenetics and epitranscriptomics, which is yet another layer of information that's important for us to understand. So the study of epigenetics is how the genome functions across cells and tissues and environments. And one way that we look at that is at not only the sequence information, but the information around that sequence, something that we call epigenetics. And so epi is a prefix that means above or upon. And so epigenetics is the study of heritable phenotypic variation, and I'll describe heritable in a little bit, but it's the study of heritable phenotypic variation that doesn't involve changes to the DNA sequence. So this is phenotypes that are distinguishable, but that's not due to genetics. And that's usually due to reversible changes to the chromosome or to an RNA molecule that affects biological function and that's often due to environmental changes. So an example of that is shown on the right, where we have these two individual mice are actually the same genotype. They have no differences in their DNA sequence, but obviously they have a difference in phenotype. And we know that it's true to the fact that if you change the maternal diet of the fat yellow individual on the left, and she's pregnant, you change her diet and the offspring will look like the individual on the right. So that's a phenotypic change that's not due to genetics, but due to an environmental interaction with the genetics. Ryan will come back in session 6 and describe even more complexity and our attempts to describe the complexity by looking at networks and systems biology in genomics and in phenotyping. And we can have biological networks that vary in scope. We can have biological networks that are trying to describe cellular events or organismal events, or even to the level of an ecosystem and how a genotype might interact with that ecosystem. And these networks are scale-free, and he'll describe that in more detail, but essentially what we're looking at here is how can we describe networks in terms of relationships among the entities in the network. And when you look at any scale of the network, down to only a few individuals or trying to describe most of the population, the properties of the network should stay the same. And finally, he'll talk a little bit about microbes and microorganisms and the microbiomes that they create, to look at the metabolic networks that are inside and outside hosts. In session 7, Ellen Goddard will join us, and she will lead a discussion of ethical and societal considerations of biotechnology and genetics. So, for example, she has suggested we will talk about these and pose these questions. For example, how did societal disinterest in traditional breeding become a global debate about genetically modified organisms? And will the technological advances that we see and we are already observing, like for example, gene editing, are we facing new opposition, or will there be more acceptance? And she'll also talk about, why are different views of different applications of the technology so different, depending on whether you're talking about humans, plants, or animals. She'll also discuss what are the ethical considerations that people have about the use of this technology, and are the views of the scientists similar to the public or not, and why not, and should they be different? And then finally, she'll talk about the role of data and science communication in how the society will end up using these new and possibly controversial technologies. And then finally, Eric Lyons will monitor or administer a final session where what we'll do is talk about current challenges and research topics. So the idea is that we will come and present some of the current work that we're doing. We'll allow participants to pose topics that they would like, where they would like additional information from that they didn't see covered in the previous 7 sessions. And there'll be an opportunity to have an open dialogue, an open discussion to try to facilitate future collaborations that people might have. So what I'd like to do now is just take a minute and see if there's any questions about the workshop structure, what we're planning to cover. before I spend about 15 minutes talking about some general fundamental concepts. And if you have a question, you could put it in the chat. I'll just stop for a few seconds and see if there are any questions at this point. There's a question that just came in, and Ryan's answered it about recordings. The recording will be available in the future. And the slides will also be posted, as far as I'm aware. My slides could be available. All right, any other questions? I think we'll just go ahead and go on. So what I'd like to do is maybe 10 minutes, 15 minutes. I'd like to present some fundamental concepts and then pose or give you a short description of what I think maybe one of the central things that we're really focused on, and that's complex genetics. So there are three major divisions in genetics. Traditional genetics, or what we might call transmission genetics, where the Mendel that you remember, and how genes and phenotypes pass from parent to offspring. The molecular genetics, which really looks at the molecules that are really underlying all these transmission of genetic information from parent to offspring. And population genetics, really one of the fundamental aspects of animal and plant breeding, where we try to change the frequency of good alleles versus bad alleles in a population. So just to very, very fundamentally or basic information, just so we're all on the same page, is we view genes as the fundamental unit of heredity. They come in multiple forms called alleles, like I mentioned before. And those genes confer phenotypes, especially those phenotypes that have a large control by genes. So a heritable phenotype, so something that can be inherited. Genetic information is in DNA and in RNA if it's an RNA virus. Genes are located on chromosomes, and they separate through processes of mitosis and meiosis. And this will be covered more in later hours. And then in addition, the central dogma, repeat that genetic information is transferred from DNA to RNA to protein, and the mutations are permanent changes in genetic material. Those variants in DNA sequence pass from cell to cell if the cells are dividing, or even from parent to offspring. And so then the last fundamental concept I'll pose here is just to give you that some traits are controlled by one or a few genes, and we often call those simple genetics, or even Mendelian genetics, because that's primarily what Gregor Mendel discovered and studied in the 19th century. However, some traits are affected by many genes and even environmental factors, and we call those complex traits or even quantitative genetics. So why do I want to emphasize this last point on simple and complex genetics? Well, the main reason is that this is probably the most important, well, not the most important, but an important concept to really understand why we need to have the Agricultural Genome to Phenome Initiative. And I'll go through that. So again, many traits have very large but few gene effects that control them. So for example, coat color is only controlled by a few genes. As you see on the left, plumage is a little bit more complex, but still has fairly simple genetics. So it's relatively simple to predict the phenotype from the genotype. It's simple to predict G from P from G. However, many genes, as I mentioned, or many traits are affected by many genes with small effects. And so it's very complex and difficult to predict the phenotype from knowing the genotype. And unfortunately, most economically important traits are complex, and they're affected by many genes. So predicting the genotype to the phenotype is very difficult, is hard. And so that's what we will focus on in AG2PI, but I do want to cover a little bit of simple genetics as an example here, and then we'll talk a little bit more about complex genetics. So simple genetics is still very important. It directly affects patient lives, for example, and it is important in animals and plants. This is an example of probably one of the more famous examples of using genetic testing to make informed medical decisions. So Angelina Jolie has a family in which there's a very high frequency of breast cancer. And so she was concerned about that. And she wrote in the Los Angeles Times that she ended up having to do a test, having a test and then doing a preventive double mastectomy, because a blood test told her that she carried a particular allele of the BRCA1 gene, or breast cancer 1 gene, that carried an estimated 87% risk of breast cancer. So the prediction is very strong that if you have that particular allele, that particular form of the gene, you have almost a 90% chance of getting this disease. And so because of that, she was able to begin to make some adjustments and change a likely future phenotype that she would end up with breast cancer. And so that single test on a single gene variant that had a very large effect was able to predict a future phenotype. Now, does that mean that the BRCA1 gene is the only gene controlling cancer? And of course not. We know that that's not the case, but there are some alleles that have such large effects that there can be very simple to predict phenotype from genotype. And there are tons of examples of this in the literature for humans. Of course, we study human genetics very deeply. And so there are many books, booklets, for example, from 23andMe, that will educate people on the effects of some of their mutations that they may find. And there's the government references that are also available. In terms of resources for G2P, in terms of simple genetics in agriculture, there are resources for known mutants with large effects on phenotype. So what we might call Mendelian or simple genetics, both in animals and in plants, where there's hundreds to thousands of variants known, where the gene, just knowing the gene will allow us to predict the phenotype. But unfortunately, as I mentioned before, most economically important phenotypes are caused by complex genetics. And so you already know some complex genetics. Human height is a good example. Height is controlled by many different genes. So if we would want to think about genetic improvement of tallness, so give me a break here. Obviously, we're not going to do that in any systematic way. But the idea is, you know something about this. If parents were interested in having basketball players, for example, you would start with good genes. You would start with tall parents. Why? Because we all know that. We know that inherited characteristics often can be recognized, and they'll be inherited from their parents. So you'd start with good genes. But is having tall genes enough? Do you expect your children the children of two tall parents to always be tall? Well, of course, we know that there are examples where that's not the case. So it's not a 100% accurate phenotype prediction, but it's strong, but it's not 100%. And so what else affects human height? Well, environment affects human height. So this is a graph of human height over the last almost 200 years. And you can see that across lots of different places in the globe, there has been an increase in human height over time. Well, why was that? It's not probably enough time for evolution to have changed the height of humans. It's very likely due to the environment, improved nutrition and improved medical technology. So we have to view height as a continuous trait because it's affected by genes as well as the environment. But on the other hand, height is still heritable, right? You're more likely to be able to see tall children from tall parents. So what does that mean? Again, it means that part of the variation can be measured as due to genetics. Part of it's environment, but a lot of it is due to genetics. So in livestock and in plants, we can do the same thing. We can predict differences in heritable traits in future offspring for anything that's a heritable trait. So for example, we can observe the outcome of many crosses and then learn to predict phenotypes based on information that we can gather both at the molecular level and at the population level. So we've done this. We've improved milk per cow. We've improved corn yield. And it's clear that we can do this, and it's possible to improve this, any phenotype, because there are clear rules that govern how genes move from the parent to the offspring, as well as how genes interact and control functions. So another way we can change traits in the future, and this will be probably pretty clear to some of you that have looked at some of the technology that's out there. We can use technology to mutate genetic material, and we can generate new phenotypes. Now, of course, that's dependent on knowing information about what you want to change, knowing information about genomics. So we need to know the exact genetic information in our animals and plants. And so here's a need for best understanding of our genomes and genetics in plants and animals. There's good successes. We can produce hornless dairy cattle. We can knock out genes that allow us to prevent viral infection in pigs. We can prevent caterpillars from eating our plants, and we can create rice that has vitamins in it to prevent blindness. In terms of changing our future, human future, we can predict outcomes by knowing the function of very specific molecules, DNA molecules, different genes, and associating DNA variants with different phenotypes that we find. And we've talked about this a bit, but you have to thinking about it from that genome to phenome perspective. You need all kinds of information, and that's really 23andMe's strengths. Basically, they collect a lot of genotypes, they collect a lot of phenotypes, and they can figure out which genotypes can predict phenotypes. And just one final point in terms of going back to the Angelina Jolie's story, it's even a little bit more complicated. Even simple genetic effects are not really so simple. She had a new scare a few years later, and she had to decide on doing more surgeries. And it was because she learned more than she had before. She learned not only that she carries the disease mutation, but she learned more biological information. In other words, links between genotype and phenotype. So she found out that she had additional information on inflammatory protein markers. Those were linked to additional cancers. And so this was more genotype to phenotype information. And because her female relatives had those cancers that were associated with these inflammatory protein markers, then she decided to have even more surgeries. And so she ends this saga that she described in 2015 by saying, knowledge is power. The more knowledge, the more information I had, and allowed me to affect my future, to predict, to do changes that will increase the better outcome. And our hypothesis is that this will be true for agriculture as well. I don't have time because we're running short, so I'll let you look at this interesting story of albinism in North American tribes. It's an example of the environment actually changing a population's phenotype. And I won't go through this, but it's there for you to read in the future. Essentially, what happened is that it's an example of what would normally be a negative phenotype increasing in a population because it was viewed positively in that particular population. So these are the questions that I have for the discussion, and I think what I'll do is stop sharing. This will be in your in the HackMD, as Ryan indicated. And what my understanding of what we'd like to do next is we'll break for about 15 minutes and allow you to be sorted into groups, and then you can discuss these questions and maybe what your answers would be, and then we'll come back again perhaps 10 or 15 minutes before the hour. The answers to the questions are shown on the right side in the HackMD, and those will remain up, so we don't have to actually get through all of this. We would like to try to keep on time, so we'll only probably describe things for about 10 minutes. So we probably won't get through all the breakout rooms, but maybe if we could start with breakout room 1 and maybe just describe maybe your answers to the questions, The first one is pretty straightforward. I think everyone got that one quite well. So maybe we could even skip question 1, but start with question 2 in breakout room 1. Do you have a recorder or reporter back?
Yeah, I'm Yufang, and I'm in breakout room 1, and looks like all my team members are shy, so I'll.
No, I'm here.
Okay, well, yeah.
Yeah.
So I'll let Shiv to say something.
Go ahead,
He-
Chief.
Yeah, the question number 2 was what is the difference between simple traits and the complex genetic traits? And maybe the simple traits are the Mendelian traits that are easy to transfer, that involves only 1 gene, and the complex traits are like quantitative traits that involves more than 2 genes. That's what I understand.
Very good. Are there other additions? I guess any breakout room anyone that would like to add to that answer? I think in our breakout room, we might have one addition maybe to that answer. In breakout room 4, they had the answer of that simple genetics often have very little effects due to the environment. So they're very, go ahead, Raziz.
Animals, especially cows, pooled is a simple trait, but milk yield is a complex trait because pooled, to be pooled or without pool, can cause just 1 or 2 genes. I don't know how many, but it's a simple trait. But milk yield, lots of gene or polygenomic effect, I guess. I give that examples.
Good. Any other breakout rooms want to add anything more?
Can I just ask a question about that simple trait is not usually influenced by environment? Does that apply to plants as well?
Yes, and I just to clarify, what we're talking about here is how easy or how accurate can a prediction of phenotype be if you know the genotype. And so that's what I really mean. For example, there are different mutations in plants that cause the lack of organelles, and at least that's something I know of here at Iowa State. And in Arabidopsis, you can have basically white plants because they can't make chlorophyll. And for example, that wouldn't matter whatever environment that plant was in. So that's what we mean, that there can often be environmental effects, but the idea is a simple trait is very easy to predict the phenotype if you know the genotype. Whereas complex traits, it's quite difficult because there's so many interacting factors. Hopefully that's clear. Anything else on question 2?
Just to add one little piece to that, the more genes there are, each of those genes can have some interaction with the environment. And so that's why it gets so hard to deal with the environment in a complex tree.
Very good. Thank you. Maybe we could go on to question 3 then. So that's sort of the heart of what people are trying to do in breeding. How can we use genetics to predict the future phenotype in a plant or animal? Is there any group that wants to tackle that one?
The statistics analytic tools, and we can do the Bayesian blotting, like more molecular part to predict that. Or like the example we have took that they found the breast cancer gene, and they just want to check if she is also carrying that allele or not. Angelina, yeah, that's a good example.
And I agree with that. How does that, I guess what I'm looking for is what's the process? How do we use genetics to try to predict a phenotype?
We are using markers, especially SNP in animals, and we are predict to phenotype Also, we have some data about phenotypes. I mean, milk yield or carcass traits. I mean, body weight or body conditions. So we can first, maybe we are simulated. I mean, with markers and with the data of phenotypes, and then we are trying to predict the future generation of animals. They have same yield or not.
Very good. Yeah, so the crux is that we try to collect data on phenotypes and genotypes and try to associate specific genotypes with specific phenotypes so that we can then predict phenotype from knowing that genotype. Anything else anyone wanted to add from their discussions? And then the last one is sort of wrapped up in the answer to the third one. But if we want to, if there are other ways that we can change phenotypes, that might be an additional discussion we could have. So how can we change phenotypes?
Breakout room 2, we had 2 answers to this. We suggested there was environmental effects which can change the expression of genes as phenotypes while not directly changing the genes themselves by sort of G by E expressions. And the second way was by changing the genes directly. So be that through crossover, mutation, or gene editing.
Very good. Exactly.
We can use CRISPR-Cas9 technology, maybe.
Mm-hmm. Other things that people wanted to add?
Yeah, group 8 came up with also breeding. It's a little bit slower, but it still changes eventually the genotype by the process of selection.
Very good.
And
Absolutely.
epigenetics.
Go ahead.
Epigenetics as well.
Yep, very good. So we'll talk about ways that you can change the phenotype through environmental effects in the epigenetics section. Any other comments to add? Great. Well, again, the answers will be in the permanently in the HackMD that you could look at in the future, and the slides and the recordings of this talk will be made available. So we're right on time. Congratulations. Everyone's being very well behaved. Do we want to take a minute break, or I'm not sure that we had that planned, but I think maybe it might be good to take a couple of minute break.
All right, well, hopefully we're all back. My name is Cliff Weil. I'll be talking for the next couple of sessions here. The first one first thing I'll talk about today are about the molecules that are involved in genetics, a little bit more about the central dogma that Chris talked about earlier and some slight variations on it, and a little bit about what we really mean by genotype and phenotype. And for those of you who have had some biology before, this may be old news, and so bear with me, but a little refresher might not be a bad thing. For those of you who are more information scientists and computer scientists, the idea here is to try and get you used to thinking about biology and about the kinds of information that you as an informatics person are most likely to see as we deal with going from genotype to phenotype. So what I'm going to do here is share my screen, and hopefully this is going to work. And I'll go into present mode. There are going to be a couple of movies in this, and what I need somebody to do is to unmute and squawk loudly if the movie part doesn't work. So here we go. Let's see what happens. Loading. All right, so you should all see my slides. Is that?
Yes.
You see that okay? Okay, good. All right, so the main points that I want to try and get across in this section are to introduce you to the idea that cells, particularly in organisms, are really busy, complicated, four-dimensional places. So there's a lot going on, and it's going on over time. The timelines are fairly short, and this goes for the things that are sort of the routine business of the cell on the one hand, and the way that organisms and cells can respond to the environment on the other. And so all of this requires some really clear instructions, really clear information, a lot of communicating and coordinating. This bit in the middle here is what we affectionately call the central dogma in biology, where DNA, which is an informational molecule, can make faithful copies of itself. That's what this arrow is. That DNA, the information from that DNA can get copied into another molecule called RNA, which is a little bit similar, but a little bit different as well. RNA, as it turns out, can copy its, there are ways that RNA can get copied from RNA as well as getting copied from DNA. The information in RNA gets then translated into a completely different language, into proteins, which use different molecules altogether to do it. And the proteins are what then go off and do most of the work in a cell. And the last point I really want to make sure that you take away from this half hour is that this idea of a genotype, which is essentially all of the information, the complete set of genetic information in a cell or in an organism, gets translated into phenotype, which is basically any outward manifestation of that genotype. So we talked about height, we talked about breast cancer, it can be eye color, it can be seed shape, it can be all kinds of things. But it doesn't have to be those kinds of visible traits alone. It can be a band on a gel, a peak in a mass spec. There are all sorts of ways that you can think about phenotype, and I want to introduce you to a little bit of that in this half hour as well. All right, so DNA, see, there's a bar of stuff on the top here. Okay, so biological information in most organisms that we know about is actually most often stored as DNA, this molecule, deoxyribonucleic acid. It's made up of 4 basic building blocks, which we call bases. Their names start with the letters A, C, G, and T, and that's how you will see them almost 99.9% of the time, as a string of A's, C's, G's, and T's. I suspect you've all seen DNA sequences at some point in your travels. You'll certainly see a couple here. Most of the time, that information is a combination of which building blocks there are and the order in which they occur. And so you can see down here in this picture on the right, there's a string of A's, C's, G's, and T's, right? And that's what I'm talking about. DNA is a double-stranded molecule, which means there are 2 strings of these A's, C's, G's, and T's, and they pair up with each other following very specific rules. And these rules are very strict. So A always has to pair with a T, and C always has to pair with a G, right? And these rules, like I said, are pretty much the strict law of a cell. There's all sorts of machinery in a cell for making sure that that rule is obeyed, and there's all sorts of machinery in a cell for recognizing when it isn't and fixing it. And the fact that these rules are so strict and are followed so faithfully is what allows a string of DNA to make a copy of itself. And that's really important. We'll talk about that in just a second, but mistakes in that can happen. So one of these bases might change, right? And if it changes, then the thing that it pairs with when things get copied might also change. And these kinds of changes, these sorts of mistakes, are mutations. And things that have mutations in them are mutants. So we generally think of DNA as being this double-stranded kind of a thing, and these pairs of bases as base pairs. And so you'll often hear about people talking about how many base pairs something has in it. And for example, humans, you have about 3.2 billion of these things in your genetic code. So how does a strict base pairing allow copying? Well, it turns out because of this double-stranded nature of things, if you peel these strands apart, like is shown here in this cartoon, and now you see that we've got sort of the A and the T are separated here, and the T and the A are separated here. I can come through here and ask, what information do I need to pair up with this A in order to make things whole again? Well, if a T always goes there and an A always goes here, by the time I get done, I have another AT pair at both positions. So I've gone from having one AT to having now two faithful copies. And you can see that kind of played out down here. The old strand of DNA is shown here in red, and the new strand that's been copied is shown here in blue. And what you can see is that the copies on either side here are exactly the same. And so this sort of base pairing rules allow us to faithfully make two new strands out of one old strand. Now, it turns out that there's a lot of DNA in any given cell. There's literally 6 feet of DNA in every single one of your cells. And nuclei are so small that you can fit 10,000 of them on the head of a pin, right? And so in order to package all of this stuff into something that small, it takes a lot of sort of winding and scrunching. And that is done by proteins. So DNA, shown here as this double-stranded molecule, gets packaged up into something that really gets crunched together multiple times to create this sort of a thing, which is a chromosome. It gets packaged up into something called chromatin. Chromatin is a combination of proteins and DNA, and all of that scrunched up chromatin, those bundles are chromosomes. And so I've got a short movie to show you here that comes from the DNA Learning Center at Cold Spring Harbor, and hopefully this will work. Can you all see the movie? Can you all see a movie screen? Hopefully you
so
can.
far, yes.
See
it's
it.
not-
Okay.
Yes.
Yeah.
Okay, awesome. All right, so let me start this. So what you have here, can you hear the audio?
No.
You can't. Okay, good. So this is the DNA molecule, and this protein, 8 different proteins are going to land on the DNA, bind together to make a complex, and wrap the DNA around them. And that compacted thing is called a nucleosome. There's about 100 base pairs around this. And these will now sort of coil on top of each other and get a fiber together that's called chromatin, all right? And at this point in time, the fiber is about 30 nanometers across. And it turns out that the DNA exists in your cells most of the time like this, wrapped up in this kind of a fiber that's about 30 nanometers across. And anything that gets in there to try and use the DNA or read the DNA has to figure out how to do it in this kind of a context. So this then gets even further packaged by even more proteins that aren't shown here into something that eventually folds up into something that lets 6 feet of DNA squeeze into each nucleus. And so what will happen here in a second, as the movie goes forward, is that you'll see this basically turning into a chromosome like what you're used to seeing pictures of. So we can see those things in microscopes. Here you can see them in a cell. This cell is about to divide. And what chromosomes allow to happen is the even and accurate distribution of all the information into both of the 2 cells that are the products of division. You can see that happening here in this movie, all right? So there's all the chromosomes getting dragged into 2 different cells, and then all those bundles kind of relax back into that form that allows the DNA to get used. All right, so let me stop that and go back to this. All right, so there's the chromosome. And if you want to go see these videos on your own time, you can pull up the slides and the link is there in the slide. Okay, so DNA is one molecule, but what then happens is that the information in the DNA is going to get transferred or transcribed into another similar kind of a molecule called RNA, ribonucleic acid, which is kind of similar to DNA, but a little bit different. It also has 4 building blocks, although one of them is different. There's something called uracil, or U, instead of thymine or T. And the other difference is that there's usually only 1 strand of RNA instead of the 2 strands of DNA. And the backbone is made up of something that's a little bit different. It's a slightly different sugar, ribose instead of deoxyribose. But the main difference that you need to know about is that it's single-stranded and that it can sort of fold itself up, where DNA doesn't do that quite so well. DNA got scrunched up by proteins. RNA can pretty much fold up all by itself. Now, it turns out there are 4 major bases for RNA, the same way there were 4 major bases for DNA. But it also turns out there's about 150 other weird variations on bases for RNA. And we think actually that RNA predated DNA. RNA was maybe the very first molecule that there was. And so it was kind of information. It could fold up and do things. It had all kinds of different functions. And so the fact that it can fold up matters. It turns out that all that folding lets it do stuff, including making double-stranded parts of itself. It can fold up on itself, something DNA can sort of do, but not very easily. So here's a picture of some folded up RNA. So the RNA is actually folding up into kinds of machines, one of which allows information to get, this is a molecule that allows proteins to get strung together. We're going to take a look at that in a second. Here's a big giant pile of RNA and protein that folds up into a machine that allows that information to get translated from RNA to protein. We'll take a look at some of those things in a minute. But the main point I want to get across here is that you can fold RNA up into stuff. All right, proteins turn out to be the last piece of this central dogma. DNA gets transcribed into RNA, and that RNA, the information of that RNA gets translated into proteins. Proteins are also chains of building blocks, just like those nucleic acids were, but this time instead of 4 of them or 150 of them, there were 20 of them. And they're a completely different kind of molecule. They're an amino acid. And the only thing that you really need to remember about this is that it's kind of a different language than DNA and RNA were. And that's why we say things get translated, because we're changing languages now. The story is a little similar in that which amino acids get strung together in what order dictate how these proteins will fold up. And it's all about how they fold up that dictates how they work. So the shape they fold into, details about their surface charge, what in the cell these proteins interact with, where they get made, when they get made, all these things are important things that dictate how these function. And ultimately, how the information that used to be in DNA is going to get interpreted into function and phenotype. And remember, we're all talking here about how genotypes become phenotypes. So these function and interactions among these proteins, and there are literally tens, if not hundreds of thousands of proteins in a cell, are important in determining what we see as traits and characteristics and phenotypes. All right, so the central dogma, how does this all get how does the information get transmitted from being DNA into being proteins? Well, it turns out that DNA gets used as a template to make RNA based on those same base pairing rules. So every time there's an A in a DNA, there's a T in an RNA that's getting made from it. Every time there's a C, there's a G. I'm sorry, every time there's an A in an RNA, an A in a DNA, there will be a U in an RNA, not a T. Every time there's a C, there will be a G. Every time there's a T, there will be an A in the RNA. And these sorts of base pairing rules end up allowing the DNA to get transcribed. So there's transcription of DNA into an RNA copy or a transcript. And you'll hear oftentimes about transcripts, lots of transcripts collected together. How many transcripts are there? What those transcripts are. There are signals in the DNA that define where and when and under what conditions a transcript will start. And what I want you to notice over here is there's DNA, there's RNA, there's protein, and there's these dotted green lines that sort of don't fit this DNA makes RNA makes protein dogma, because it turns out that sometimes RNA gets reverse transcribed into DNA, and sometimes you can make RNA starting with RNA. You don't have to start with DNA. So the central dogma has been improved, annotated, decorated, expanded in the last number of years. And as you, as information scientists, will start to think about how information is getting used and generated, you'll hear about some of these things: reverse transcription, RNA being used to make more RNA. Now, it turns out that only 1% of all the RNA that gets made from DNA gets made into what's called a messenger RNA or mRNA, and you'll hear that term tossed about a lot too. And that mRNA is going to get translated 3 letters at a time into protein following a genetic code. All right. Every possible combination of 3 bases means add a particular amino acid. And we'll take a look now at a movie, and just let me preview this movie by telling you what you're going to see is DNA and an RNA copy of a piece of that DNA getting made. And at the very beginning of the movie, a bunch of things are all going to be gathering together on a particular part of the DNA. And those are all a bunch of different proteins that are guided to that place based on the sequence of the DNA and the proteins that bind there first. So there's a big machine that gets built, and at the very last minute, you're going to see one protein that got bound someplace else on the DNA kind of swoop in and touch the complex and start the transcription process. And we'll see that right now. All right, so there's DNA again. And there's that complex beginning to form. So a lot of things are going to gather at this spot. More and more and more of them add on in a special sequence, and then in comes that last one, and we're off. Now, the thing I want you to notice in this movie is that this is as fast as this really happens. That DNA is getting copied into RNA just this fast. So new bases to make RNA are flying in this intake hole, and everything is getting copied by those base pairing rules to make a piece of RNA. Now, what you'll notice is, didn't mean to stop that. Sorry. Oh, really? No. There we go. All right, what you'll notice is the color kind of changed here, and part of this RNA has become green. Nothing's different. That's still all the same bases that were getting made before. But this particular part of the RNA is going to do something weird. The cell is going to actually remove it. It's going to splice it out of this RNA and throw it away. Why are these things here? We're not entirely sure. Sometimes they have important regulatory pieces of information in them. Sometimes they have particular structures that are necessary. We're not entirely sure what they are, but we call these pieces that are going to get kicked out introns or intervening sequences. And what you'll see next is a machinery is going to assemble on this particular green part, and in a very coordinated effort, very carefully rip it out of the RNA and glue the 2 yellow ends that you see on the left and the right here together. So in come all of these molecules that will recognize special signals on the RNA and gradually pull the 2 yellow parts on the left and the right together, put them together, and cut out this piece of green RNA, right? And what's interesting is in this RNA, there are a lot of these introns. There isn't just one. There can be 15, 20, 30 in some cases. And gradually, all of the yellow pieces will get stitched together into the finished product, which is the thing that's going to get translated into a protein. So here you can see a bunch of these green things getting spliced out. And this all happens in the nucleus of a cell where this thing got transcribed to begin with. And only after it's all finished, when all the yellow parts have been stitched together, will this now get used to translate into a protein. And it's going to leave the nucleus to do that. So it's going to go out of the RNA into the cytoplasm of the cell. And now a machine is going to assemble on this RNA and begin to read it. And it's going to read it 3 bases at a time. And each of those 3 bases means insert an amino acid. And that we'll take a look at a second about how that's done. But this is going to read along 3 bases at a time. So there's the rest of the machine. And it starts clicking along here, 3 at a time. These green triangles that are flying around here, each have an amino acid attached to them that's going to go into the protein, and a sequence on the other end of them that matches, by base pairing rules, the 3 bases of the RNA that are in the machine here. It happens about the speed you're seeing it here. So it's a pretty amazing thing if you think about that all of these things can be happening as fast as they're happening and as accurately as they happen. The protein, which is kind of emerging from the outside of this ribosome here, folds up as it comes out into its finished product. All right. Let me get rid of that. Sorry, it's still going. All right. So how does it know how to do this? As I said, there are 3 bases that are read at a time, and each 3 bases corresponds to insert a particular amino acid now. And it does that by following a code. So here are all the possible 3-letter combinations of RNA that there are, and the amino acids that they correspond to. The thing that you really just need to take away from all of this mess, since we're running a little low on time here, is that there has to be a particular organization to those 3 letters. And what I've shown you here are basically a sentence that makes sense if you read the 3 letters in the order that they're shown here in the sequence, these 3 letters at a time, the, big, cat, etc. If I take those exact same letters and I change the reading frame, in other words, I shift everything over by 1, it doesn't make any sense. That's all gibberish. In fact, if I shift over another one, it's still all gibberish. There's only one reading frame in which these 3 letters make some sense. The reason that matters is that there are 3 particular combinations that say, stop making a protein now. And if you run into those 3, then you stop making a protein. So what you want to make sure is that you put amino acids together in a series that doesn't run into these stop codons until you're done, all right, until you get to the end of a protein. Each one of these things is called a codon. All right. If you get out of that reading frame, you very quickly run into a stop. And so what you have to be careful of is that the 3 letters that you're reading, so here's 3 letters that mean this amino acid, 3 letters that mean this amino acid, 3 letters that mean this amino acid, etc. Eventually you run into a stop. But if you stay in the reading frame that is in the green line here, you don't run into a stop, right? If you add a base, you switch the reading frame, as I've shown you here. So if I stuck an extra letter into the big cat ate the fat rat, as soon as I stick in that extra letter, I mess everything up afterwards, all right? So what's important as you're looking at genes is to look for places where transcripts can start and get made, and places within those transcripts where you have a start signal for a protein, a long series of things without a stop, and then a stop. All right, those are the characteristics that will describe a gene. All right, so a place for transcription to start and a place for transcription to stop, a place for translation to start and a place for translation to stop. Introns get taken out, leaving the exons behind, and all of those constitute things that make a gene. And as information scientists, will look at long strings of DNA and ask, where are the genes? What you'll look for are those signals as a collection. So the signal for a transcript to start, within it, a transcript that has a start for translation and a stop for translation, and a signal for the transcript to stop. Those things all taken together will be a signal to a computer program or to you that you might be looking at a piece of DNA that encodes a gene. All the transcripts, all the genes taken together are referred to as the genome. All the transcripts together are the transcriptome. All the proteins together are the proteome. All the metabolites together are the metabolome. All the thundards together are Tina Turner coming after you with a crossbow. There are also things that are called pangenomes that you'll see oftentimes, especially as you all go forward. And these are places where all the genes that we think are in an organism are in the particular organism that we looked at. If I go to a different variety of the same organism, so a different variety of a corn plant or a different breed of cow, what I'll find is that all the genes that were in the first one are not necessarily in the second one. And so if I want to think of how many genes, what are all the genes that are in corn? I need to think about what all the genes are in all kinds of different corn. And those things all taken together are what are referred to as a pangenome. All right, the last thing I'm going to tell you about, and I'll do this really quickly, is to put a slightly different spin on what Chris was talking about when he was talking about epigenetics and an epigenome. What I want you to think about as you think about how epigenetics works, and there will be a whole session on this later, is that epigenetics also has something to say about how genes get used, not just whether they get used, but whether the cell can even see that they're there to use at all. And so the best way I have to think about that, I think, is to consider these sentences down here at the bottom. So we're going to ask not only which sentences get read together, but whether they're even readable by a cell. So here's 3 sentences. The sun rose gently in the sky. The horses grazed peacefully in the meadow. The smoke rose gently into the sky. And that conjures in your mind a picture, all right? But what can happen in a cell is that another sentence, which is there right now, but you just can't read it, might be revealed in a different cell. So the sun rose gently in the sky. The cow knocked over the lantern and the house burned to the ground. The smoke rose gently into the sky. That's a different picture, right? All the same 4 sentences were here, but you couldn't read one of them in the first idea, and you couldn't read one of them in the second idea, and it changed your picture. And so this is sometimes how cells can change their identity, right? The difference between a brain cell and a liver cell. How does the brain cell know it's a brain cell? Some of the genes in it get read, and some of the genes in it can't be seen by the cell to read. In the liver cell, other genes can't be read, right? And so this collection of information, what information is available to you to read, is a lot of how epigenetics works. And those things can change, and you'll hear more about that later. All right, last thing. Cells are busy, crowded places, right? Lots of things going on. We're used to seeing pictures of cells as having lots of empty space in them. But the truth is not that simple. The truth is more like what's over here on the right. Lots of things all crammed together. And better yet, this picture on the bottom. That's a picture of a street and all those people. And what I want you to imagine in a cell, and this is really what goes on, is that you have to get from the lower right corner of that picture to the upper left corner of that picture. You have to do it in a fraction of a second. You have to talk to 6 specific people on the way, and you have to do very specific things with those 6 people, and you have to get to the other corner and hand something to the person that you meet. It's that complicated inside a cell, and it happens that fast. And what you'll see going on in a number of species now is the attempt to form what's called a cell atlas, which is to understand where everything is in a cell and when it's there. Because we don't even know what all these people are, who all these people are, and what all those people are doing. And we're still trying to figure that out. All right. Oh, I did have one more thing to say. Genotype and phenotype. So genotype is the collective genetic information in an organism. Phenotype is any outward manifestation of that genotype. Could be eye color, could be seeds, could be tail length. But here's a gene that if you change it, can change the male part of a corn plant into a female part of a corn plant, all right? Phenotype can be all sorts of other things, biochemical compounds, behaviors, peaks on gels, all that sort of thing. And the last thing, and we talked about this earlier, phenotype can result from the interaction of genotype with the environment. The environment can have a huge role to play in a phenotype. And what you see here, and I think you probably all did these experiments when you were in grade school, Here are two genetically identical plants. One germinated in the light, one germinated in the dark. You can see a very distinct difference in the phenotypes here based solely on this one difference in their environment. All right, now I think I'm done talking. All right, so there are the 3 points that I wanted you to take away from all of this. I won't go through them again because we're way over time, but I will stop sharing and we can go to some breakout rooms and talk about the questions. All right, we're back. So let's come up here to the beginning and start with question 1. Describe an example that was not the little plant that I showed you at the very end. That is an example of an identical gene, two identical organisms with two identical genotypes producing different phenotypes. So a lot of you had twins. Good. That's good. How many of you know sets of twins? Well, I can't see anybody's hand, so I guess that's a silly question to ask. But you probably, anybody who does know sets of twins? Lots of thumbs, okay. You realize you can tell the two twins apart. They really aren't exactly the same, right? Even though they're genetically the same. So that's one example. Chris showed you another example of the mice, right? The fat yellow mouse and the thinner wild type looking mouse. Twin sheep reared in different environments, somebody said. Good. Yeah, so all right, so I think you guys have the sense of what those were. So let's move on to question 2. Which do you think might have a more severe effect on a gene? Changing a single base from a G to an A or removing the G altogether? Anybody want to leap in and tell us what your group thought? Group 2, here, you said you were unsure.
Cliff, this is group 3. We had 2 different answers.
Okay.
One of us felt that it would mess up the codon, so it would have the same effects.
Mm-hmm.
But one of our participants also felt that probably dropping it would have less effect than changing it.
Taking one out would have less of an effect?
Yes.
Okay.
She said because when she's a computer scientist working with gene data sets, and many times the sequences go missing is what her experience says.
Okay, but what happens if you take a single base out of a gene that you're trying to translate into the big cat ate the fat rat?
This is group 9 here. We thought that a deletion would actually be far more severe, because if you're thinking about it in a reading frame where you have, you're reading them 3 base pairs to a codon, a deletion of one of the amino acids will cause a shift for all the codons further down. So everything is everything is going to get jumbled up. Whereas if you just changed one, in an amino acid, the amino acid would change, but all the ones subsequent would not be affected, whereas the removal of one affects all of the ones downstream.
I think that's a really good answer. And I think that's probably the best answer, in that you might change something if you change the G to an A. You might change something into a stop, and that would be pretty severe, but you might not. You might change it into something that's pretty similar, or allows a very similar amino acid to be incorporated. So you might have a severe effect, you might not. But if you take that base out, just like Samuel said, you change the reading frame, and that's going to be pretty much a disaster. So that's the best answer there.
Cliff, I have a question, if that's okay.
Absolutely.
This is group 2. We were unsure because we thought that deleting a single base might allow the molecular construction mechanisms to notice there's an error, and then it might end in them deleting that copy, which wouldn't be so bad because in the end the phenotype would be unchanged.
Fair enough, but a lot of times if you delete a base, the machinery has already sort of come through and decided that that deleted base was something it just dealt with. And now the sequence just has the missing base. So this, I might have been a little clearer here. This is if something like that happens, if a base changes or if a base is deleted, the machinery has already kind of processed that and dealt with it. And now you're left with the result. So that's my fault for giving you an unclear question.
But I think I could add to that, Cliff, exactly what you're saying, and maybe the fact that most of the error correction machinery is at the level of incorrect base pairing. If the DNA, if the DNA is intact, that the base has just been removed on both sides, then it's going to look like a normal DNA. If the base is deleted only on one strand, then yes, there could be a repair. But the problem is that often the machinery doesn't know which strand is right, right? So it sometimes will correct and remove the base from the other strand. So it's not always clear from the machinery when you have insertions and deletions.
Who was the right thing and who was the wrong thing? Right? Does that make sense, Kenneth? Okay.
Yes, thank you.
All right, so let's look at question 3. This one is more for the information scientists among you to play with, but it's kind of fun to think about anyway. And this is actually real. I didn't make this up. People have synthesized 4 completely different bases, right, than the ones that I told you about and the ones that nature has invented. And they have base pairing rules, very strict base pairing rules. So what do you think might happen if we had an 8-base DNA code instead of a 4-base DNA code? So I see answers about more information and variability. Good. Breakout room 3, that's an interesting answer. It will become very complicated to predict outcomes, more phenotypes. Could you tell us more about what you meant there? Anybody from breakout room 3?
So Cliff, let me volunteer again.
Okay.
So basically the thought was it's permutation combination. And if with 4, we are dealing with this complexity, if there were 8, it would be much more complex. You have a longer sequence and more combinations to deal with, is what my thought was on it.
Okay, breakout room 4, you actually put some numbers to that. Why don't you tell us about that?
I think actually I did that. It's only justified by why. So we hypothesized with the team that probably you're going to end up with a much larger codon table, obviously, because you have more combination of bases. And if the codon system remains the same, that table might actually get much larger, something in the ballpark figure of those 2 numbers that I just mentioned in the answer.
Right, so now you have 8 to the 3rd power possible combinations. You could encode all kinds of information if you wanted to do them 3 at a time.
Indeed, yep.
All right.
Octoploids are already hard enough to work with.
Fair enough.
I'll second that answer.
Octoploids, for people who are wondering, are things that don't have 2 copies of every chromosome. They have 8 copies of every chromosome. All right. So in the interest of time, let's move to question 4, where I said gene annotation software. This is computer programs that look at DNA sequence and try and guess where the genes are. Do that on the basis of looking for a collection of the signals that I mentioned to you are involved in figuring out where genes are. A place for transcription to start, for all those proteins to assemble and start. A place for that transcription to stop. A codon in the middle where an amino acid sequence could start and translation could go to a stop codon. And it looks for that collection of things and says, this is probably a gene, and spits that information out. And what I asked here was, what do you think the pros and the cons are for that? So let's start with breakout room 1. Anybody from breakout room 1 want to talk about your answer? You're all shy.
Yeah, hi, I'm from Breakout Room 1.
Okay.
Yeah, so what we understood from the question was that like that you would feed the DNA sequence to the annotation software and like it'll tell you if it finds any de novo genes in it. So I thought like the pros of the approach would be like it's a straightforward approach, like it already has like a specified DNA sequence characteristics that it looks for. And if something does not follow that order, it'll classify it as genes de novo. So it's pretty straightforward. And once you have that data already there, it's easy to find new characteristics in the sequence. The cons would be like, first, obviously, it would be, it would take a lot of time. Like the complexity would be big for this approach. And also like the software has to be constantly updated to make sure like nothing gets left out and it does not become obsolete, because I think the sequence characteristics would be like sort of specified and hard coded in it.
Fair enough. And you're also very dependent on how accurate your DNA sequence is.
Oh yeah, I mean, we have to make sure that it is like correctly assembled.
I'm gonna scroll down here to breakout room number 8. That's a very interesting answer. Anybody from breakout room 8 want to talk about what you thought?
Well, I will speak up.
Okay.
So obviously it's a great tool to detect genes in a genome database, but there's a number of issues you have to take into consideration. You need a lot more than just a start codon. You need to understand where the introns are, so you know what the sequence gets translated into. Regulatory genes don't necessarily look the same as structural genes, and are, transcription factors are a lot harder to identify. And various genetic artifacts that we don't even know what the purpose is yet.
Okay, and I think that gets to, since we're kind of out of time here, I think that gets to a really important point, which is that eventually you're going to have to do some experiments on these things to figure out whether or not they're genes, or whether the gene was correctly called by the computer. So computer algorithm will have to struggle for the same reasons that David just articulated, absolutely. And even if the computer gets everything what looks like right, you still have to do biology experiments testing whether or not genes actually are where you think they are. And we'll talk a little bit next time about how all that works. So with that, I'm going to turn it back over to Nicole, since we're at what is 3 o'clock for her.
Yes, it is 3 o'clock Central Time. So that will be the end of this session. We hope to see you in the next 3 Thursdays for our next sessions. Ryan, did you have anything to add?
Nope. Just I messaged everybody in the chat. So the right access is now closed to the HackMDs from today, and we'll be using those for next week as well. New ones, not the same ones.
Okay, great. And we will share those links to you next time. If anyone experienced any problems or have any feedback, feel free to email me. I will put my email in the chat now. And otherwise, have a great rest of the day.
Thank you all for coming.